Hello friends, welcome back to the journey of microbiology. We are discussing the immunity chapter in microbiology, in which we discussed about the immunology, what is meant by the immunology and their branches. We also studied that immunity is something which resists the pathogenic growth in our body as well as the other specific antigens, the foreign particles, cellular debris, allergens, etc., and etc. Okay. Then we studied that immunity is come under the system, and that system is called the immune system. Okay, that system is called the immune system. Then we divided the immune systems into the two parts based on their origination or origin. We divided immunity into innate or inborn immunity, also called as non-specific type of immunity, and we divide the another part of immunity to the acquired immunity, also called as specific or adaptive immunity. Okay, so now first we will see our lecture outline, and then we will start discussing about the T cell. Okay, T cell, which is a kind of lymphocyte, a kind of basically the adaptive or acquired immunity, their production, their maturations and their functions. Okay, so first see the lecture outline. We already studied about the meaning of immunology. We also studied that immunity and their types. Then today topic we will discuss about the development and processing of lymphocyte. Okay, now see the board. Development and processing of lymphocyte. Development and processing of lymphocyte. Lymphocytes. Clear? We'll discuss the development and processing of lymphocyte. So before describing about the lymphocyte, I want to know about something from your side. Did you first heard about this term lymphocytes? Yeah, it's a kind of WBC. It's a type of WBC which produce from, very good, which produce from the bone marrow. Okay, primarily which found in the bone marrow, and for their further specializations, further prolifications, maturations, they have to come in different organs. Okay, and once they become mature or formed, they goes to the storage organ, and those storage organ are, very good, the lymphoid tissue, or we can say the lymphoid organs. What are the lymphoid organs? Yeah, lymphoid organs are classified into the two types. First one is called the primary lymphoid organ, and second one is called the secondary lymphoid organ. What are the primary lymphoid organ? Yeah, primary lymphoid organs are basically the thymus. Okay, and second thing is what? The first thing is called thymus, and the second storage house of this lymphocyte is bone marrow. Second is the bone marrow. Then we have the secondary lymphocytes. Secondary lymphoid organs. What are the secondary lymphoid organs? Yeah, the first one is spleen, second one is MALT, that is mucosa associated lymphoid tissue, which is present in our GIT and in our throat area, as a pear patch. Okay, in the form of tonsil, different types of tonsils. Clear? Now you know that lymphocyte is classified into the two types. What are these two types? The first is T lymphocyte and second is the B lymphocyte. Okay, let me summarize that lymphocytes are the types of WBC or type of leukocyte, which formed primarily in the bone marrow and stored in the primary or secondary lymphoid organs. Clear? Then lymphocytes are two types, T lymphocyte and B lymphocyte. These T and B lymphocyte constitute which part of immune system? Very good. It constitute the acquired. Immunity. Acquired immunity, which is also called specific immunity. Clear? Now we have the basic idea about the lymphocyte. So now we will take first the T cell. Okay, first take the T cells and describe it in detail. So now we are discussing about the development and maturation of T lymphocyte. Which lymphocyte? T lymphocyte. First I want to know from you guys that where they are originated. They originated from the bone marrow house. Suppose this is a bone marrow. Okay, this is a bone marrow, and bone marrow are present in the long bones. Okay, bone marrow are present in the long bones. This is a bone marrow house. Okay, so before the bone marrow, like we are not directly born as in the form of we have the bones, legs, feet, everything. Okay, we first come from the embryo. Okay, when the egg and sperm, they fuse with each other, they form the one diploid structure, and that diploid structure is called as, very good, zygote. That diploid structure is called zygote. Clear? Now these zygote, they eventually, by many multiplication divisions, they form the embryo. Clear? They form the embryo. And these embryo, okay, in this embryo, there are the precursor cells. Which precursor cells? Very good. Hemopoietic precursor cell. They are the hemopoietic, or we can say the cells which form the blood components. They are called the hemopoietic cells or hemoprecursor cells. Precursor cell. Clear? Hemoprecursor cells. They are the hemoprecursor cells. See, now in the process of development of the embryo, okay, there is formation, there is organogenesis, means there is a formation of organ. Okay, there is a formation of organ. Suppose this is one premature form of fetus. Okay, the form of fetus, and that there is a developing liver along with the spleen. There is a liver and the spleen. So these heme precursor cells, they migrate, okay, during the intra-embryonic life, they migrate into the liver. Okay, and after the birth, these precursor cells, they come into the bone marrow house. When the bone is formed, these precursor cells, they come into the bones. Clear? They comes into the bones. And then what happened? These cells, they are known as the pluripotent stem cell. Pluripotent. Suppose, okay, I write the name plurio. Write down this name. Pluripotent stem cell. First, understand the meaning of this pluripotent stem cell. Pluripotent stem cell means the cell which has the capacity to form the entire body, we can say the entire organ, okay, and in each time when they are divided, one cell is kept preserved and another cells goes into the divisions. Like for example, see, this is one pluripotent cells. When it's multiply into the two parts, one part again goes back and this part further divide, and again one part preserved, second part further divide. This is a tendency of a stem cell. This is a tendency of the stem cells, and this is a one type of stem cell, means pluripotent. Pluro means the whole. Pluro means whole. Pluro means the whole cell. Okay, we can say the many cells. It's the pluripotent stem cell which forms the blood cells. Now, understanding the meaning of pluripotent cells, now we move ahead and understand the formation of these cells. Okay, formation of these cells. Clear? Okay. So these stem cells has very unique functions. Since nowadays there is a different kind of stem cell bank where stem cells are preserved for further use. When the organ failures happen, you can produce a new organogenesis can be happened by the use of the precursors, or we can say the stem cells. Okay, so these stem cells, they divided into the two types. The first type is called as the lymphoid progenitor cell. The first type is called as the lymphoid progenitor cell. And another one is called the myeloid progenitor cell. Myeloid progenitor cells and lymphoid progenitor cells. These lymphoid progenitor cell will further goes where they goes to the one bilobed structure. Okay, one bilobed structure, and that bilobed structure is called as, very good, thymus. First understand, these lymphoid progenitor cell, they divide into the three types. Okay, the first one is called the T cell, second one is called the B cell, and third category is called the natural killer cell. These all three cells are basically the immune cell. All these three cells are the immune cell which have functions in the maintenance of our immune functions. Okay, immunity functions, they are involved in the immune functions. So among these cells, the T lymphocyte goes to the thymus. Okay, and here by the activity of thymosin, or we can say the thymus releasing factors, by the help of thymus releasing factors, these premature, we can say, or unprocessed T lymphocyte got processed and becomes the developed T cell. Okay, let me summarize it once again. We have sperm and egg. When they got fertilized, they form the one multicellular structure or diploid structure. No problem. That diploid structure is called as zygote. That diploid structure is called as zygote. Now this zygote, after the subsequent divisions, okay, they form the embryo. And in the embryo there is a expression of gene, and those genes form the hemoprecursor cells. And these hemoprecursor cells, when the maturation happens, when the organogenesis start, they migrate to the fetus liver. Okay, and in the fetus liver, after the birth, they went into the bone marrow. And in the bone marrow, these cells, what are the cells? These cells are called the, very good, pluripotent stem cells. These pluripotent stem cells, they divided into the two types. The first type is called as the lymphoid progenitor cells, and another one is called the myeloid progenitor cells. Lymphoid progenitor cells, they express into the three types of cells. The first cell is called the T cell, second is called natural killer cells, and third one is called the B cell. And if you really want to know about these myeloid progenitor cells, they form the rest of the WBC, like the eosinophil, basophil, neutrophils, and also our RBC. Don't forget, also produce the platelets. Okay, so these are the one classification. Now these T cells, NK cell or B cell which formed newly, they are not functional. They are not functional. Okay, they are not functional. So they have to be functional in order to destroy the bacteria, fight with the pathogens. They have to bear some of the specialized, we can say, the structure, or in the more specifically call it the receptors, so that they can recognize the virus, bacteria, or any other things, antigen. Okay, so for that purpose this T cell migrate into the thymus gland. Thymus gland is basically a bilobed structure. Thymus gland is a bilobed structure which present behind the media sternum, media sternum area. Clear? Now, after understanding the basic embryology of these cells, the blood components, now we move to a study detail about the T lymphocytes, maturation and purification. So first I draw one diagram. First I draw the diagram of the bone marrow once again. See, this is a bone marrow house. Okay, this is a bone marrow, and in the bone marrow, as you know, understood now, that there is something called the pluripotent stem cell. Okay, they divided into the two types. First type is called as the lymphoid progenitor cell, second type is called the myeloid progenitor cells. And these lymphoid progenitor cells, they form the three types of cell. The first type is called T cell, then NK cell, and natural killer cells, and then the B lymphocyte. Now we understood till now that this T lymphocyte need to be processed, need to be developed in order to bear some of the specialized structure. Okay, so for that very purpose, these T lymphocytes, they goes to the thymus or one bilobed structure. See, this is one functional diagram of, it's not an exact anatomical diagram, that's one functional diagram of the thymus gland. Okay, thymus have two parts. Thymus have the two parts. The first part is called cortex. First part is called the cortex, and the innermost part, which is the dense part, this part is called the medulla. This part is called the medullary part. So in the cortical region, these T lymphocyte first comes. Okay, these T lymphocyte first come into the cortical, or we can say the cortex region. Okay, now they are in the form of, these are the septa. These are the septa, and many T lymphocytes through the circulation. You know, there is circulation. So first these cells go into our circulation, the blood circulation, and then they goes to the this thymus gland. Now what happened when they goes to the thymus gland? Thymus gland have cells. Thymus gland have the cell. Write down the name of those cells. The first cell is called as epithelial cells. First cell is called as the epithelial cells, and the another cell is called as the lymphocytes. First one is called as the epitheliocyte and second one is called the lymphocyte. Write down the word. First is called the epitheliocyte, and another one is called as the lymphocytes. They are different than these lymphocytes, okay, not confused with this. So these epithelial and lymphocyte, what they are doing, they are secreting some of the substances. Like they are secreting the thymosin, okay, these thymosins are involved in the maturation. Cells are involved in the maturations. They also secrete the thymopoietin. Okay, thymopoietin. These thymosin and thymoproteins, they are acting on these cells. What are these cells? T lymphocytes, and make them mature and stored into the medulla. Okay, and here are so many immature, we can say the T lymphocyte, which are under the effect of thymosin and thymopoietin. They are maturing themselves. Clear? Okay, so after the maturations, they have again go back to the circulation. Okay, it's a mature T lymphocyte, or we can say the T cell. Okay, they are the mature T lymphocyte and T cells, and after going into the circulation, they are stored in the lymphoid organs. And what are those lymphoid organs? We know that they are the spleen. Okay, they are the spleen and bone marrow. Okay, also the secondary organs like the malt, mucosa associated lymphoid tissue. Okay, and the another one we have the lymph nodes. Okay, so that's the whole story of the production and maturation of this T lymphocyte. Okay, let me summarize it once again. You know, from our intrauterine life, when the zygote start to developing, they develop into the various stages, then they migrate into the bone marrow, and in the bone marrow these cells is called as the pluripotent stem cells. These pluripotent stem cells, they divided into the two types. The first type is the lymphoid progenitor cells, another one is called the myeloid progenitor cell. From the lymphoid progenitor cells, three cells are produced: T lymphocyte, natural killer cells, and the B cells. Okay, and in the myeloid progenitor cells, four types of cells is produced, that's the RBC and rest of the WBC as well as the platelets. Okay, now this T lymphocyte, newly formed T lymphocyte, they have to be matured in order to destroy the microbes, develop some of the specialized structures. For that purpose, these T lymphocytes migrate into the thymus. In the thymus, there are the two parts. The first part, the outermost part is called the cortex, innermost part is called the medulla, and these cortex, they are lined by the some of the cells. The first cell is called the epitheliocyte, and the another cells is called as the lymphocyte. These epitheliocytes, they secreting some of the substances like such as thymosin and thymopectins. These thymosin and thymopectins, they are working on the T lymphocytes, making them processed, making them specialized, and sending into the medullary region. In the medullary regions, they again migrate into the circulation, and once they reach into the circulation, they have to be stored, and for the storage purpose, there are some of the called the primary lymphoid organs, some of the called the secondary lymphoid organ, and where they are stored. Okay, where they are stored. That's the one basic outline of the development and processing of the T lymphocyte. Okay, about the T lymphocyte. Now, as we understood, okay, these cells, they are formed, but they formed in which manner? What happened to these cells?
how they got mature. So for their maturation purpose, we divided into them into the 4 events. Okay, we divided into them into the 4 events. Okay, the first event is related with their surface modification, T lymphocyte surface modification. Okay, in this surface modification, what happened under the work of T lymphocyte, the polypeptide chain present on the T cell, okay, they make one receptor. See, suppose this is one T cell, okay, this is one T cell. Okay, now if this is a cell, they must have one very good nucleus, which is the main program things which program all the cellular functions, cellular structure. So what happened under the effect of 2 hormones? First one is called the thymosin and thymopoietin. There are others also, but they are not more relevant, so I am not discussing. Just taking these 2, thymosin and thymopoietins, they work on the nucleus. Basically, they direct the nucleus to synthesize some of the receptors on their outer membrane. Okay, or in more specifically, we can say they direct the nucleus, and inside the nucleus, what things are there? The genes. Okay, they direct the genes to synthesize some of the receptors. Okay, or basically they rearrange their surface proteins, their surface polypeptide chain. Okay, their surface polypeptide chain. Okay, let me tell you once again. This is one gene, okay, present in the T cell under the effect of thymosin, which is secreted by what? Very good. The thymus gland. Under the effect of thymosin, they synthesize the mRNA, okay, and that mRNA result into some of the proteins, and those proteins is called the T cell receptor. What they are called? T cell receptor, TCR. Understood? First thing is immature T cell coming from the bone marrow house. Okay, under the effect of thymosin and what? Thymopoietin. Where? In the thymus cortex area, they are directed the nucleus to synthesize mRNA for synthesizing the new proteins, and that proteins form this T lymphocyte cell receptor. Clear? Now that's the first event. Okay, mark first event. Okay, I am writing event here. First event. What happened in the first event? There is a TCR receptor formation. Okay, TCR means T lymphocyte cell receptor formation. Clear? Now, in the second event, there is a selection of the beta chain. Okay, there is a selection of beta chain in the TCR. As you know that there is a sequence of amino acid in the proteins is made up of the polypeptide chain. Okay, suppose these are the one amino acid, this is the second, they are linked with one peptide chains, and there are so many chains which is called polypeptide chain. Okay, these polypeptide chains, there is the arrangement of alpha and beta chain. There is presence of alpha and the beta chains. Okay, so for making the TCR, that receptor functional, their alpha chain have to be eliminated, or we can say destruction. Alpha cell destruction is needed. Okay, in maturation phase. So in the second event, what happened? There is a selection of Second event is marked by the selection of beta chain. Okay, and those cells where the beta cells is not found, they are eliminated by the process of apoptosis. Apoptosis is basically the programmed cell death. Okay, apoptosis. So here the apoptosis process happened. Apoptosis happened, and by the apoptosis these B cells are selected. These B cells are the selected. That's called the beta chain selection. That's the second event. In the third event, there is a development of double positive and double negative charge on the cell. Means these selected B chain on their TCR surface, they are forming the charges. Okay, in the third event what happened, there is a double positive and double negative charge development. Okay, double positive and double negative charge are developed on the surface of T cell. Okay, they are called as CD4 and CD8. They are called as the CD4 and CD8 and C25 and CD44. Write down CD4 and CD8. Okay, then we have CD44 and CD24. Understood now? What happened in the first event? There is a expression of some of the genes which result in the formation of T lymphocyte cell receptor. Okay, clear? Now in the second process, already the receptors are found, but these receptors, they are not mature. Okay, these receptors, they are not mature. So for their maturation purpose, there is an interaction with some of the antigens. Okay, and due to the interaction of the antigens, these receptors, they eliminate their alpha chain, because their alpha chain is no more functional in destroying the antigens. So there is undesirable. They are the undesirable. They are not desired for destroying the antigens. So there is only selection of beta chains. Okay, so whatever cells which have the beta chains, they are selected, and another cell is die by the process of apoptosis. Apoptosis means programmed cell death. Programmed cell death means what? Means now this cell is no more functional. They are not useful. So cell will itself decide that we have to die. Okay, we have not to make the overproduction as human are doing. Okay, so it's the like the apoptosis. Clear now? In the third event, what happened? These receptors, they are forming different type of charges. Okay, and basically in the form of 4 charges. We are calling these charges as the double positive or either double negative. Okay, double positive or the double negative. Now in the fourth event, what happened? Positive selection and negative selection of these charges. Positive selection and negative selection of the charges. Mark the fourth event. Now you are thinking why I am teaching it in the detail. There is a significance. I will tell you in a few seconds. I will tell you. In the fourth event, what happened? There is a selection of what? There is a selection of there is a selection of positive selection and negative selection. What have selection? Positive selection and negative selection. Positive selection and negative selection. These 4 cells, okay, they all are not functional, and our cell knows if something is not useful, our cell eliminate that part, only accept those parts which are useful, or more specifically which are functional. Okay, which are functional. So this CD44 and CD24, they are undesirable. Okay, these receptors CD44 and CD24 receptors, they are not useful. Only CD4 and CD8 they are useful. Clear? That's called the positive and negative selection of the cells. Positive and the negative selection are selection of the cells. Okay, now you heard about the autoimmune diseases. Okay, so during this development process, during this selection process, some of our cells, okay, some of our cells, these CD4 and CD24, they come back, like we can say the exact explanation, scientific explanation, but that's the belief that CD44 and CD24 cells, these cells, they are not eliminated, or we can say that they are not apoptized themselves. Either they will start destroying our own cells, okay. That's one aspect related with the autoimmunity. Okay, now the final selection. The final selection is marked by the fifth event, in which there is only single positive cell selection. Okay, in this they express only the molecules on their surface either as a CD4 and CD8. Now the fifth event is marked by the single positive cell selection. Single positive cell selection. Now only this CD4 and CD8 cells, they are present on the T cell receptor. Understood? Now let me summarize it once again. What happened during our intrauterine life? Okay, during our intrauterine life, we have some of the lymphoid tissue. Okay, we can say the hemopoietic cells which derives from the embryo, and they goes to the premature liver. And then when the birth happened, when the organogenesis, organ developments occur, they migrate into the bone marrow site, where they are known as the pluripotent stem cell. Those pluripotent stem cells, they divided into the two parts. The first type is involved in the production of T cell, B cell, or natural killer cell. That's called as the lymphoid progenitor cells, and another one, they are called the myeloid progenitor cell. Myeloid progenitor cells is basically concerned with the formation of the RBC, the rest of the WBC, that's called the granulocyte, like eosinophil, basophil, neutrophil, and the platelets. Okay, now the T cell, which is formed by the lymphoid progenitor cells, they are not matured. They need to be processed by the thymus gland. Okay, so they migrate to the thymus gland, and in the thymus gland, under the secretion of some of the thymus, that's called the hemopoietin or thymosin, they start developing the receptor, that's called their gene are expressed. The T cells genes are directed by the thymosin, which result in the formation of T cell receptor. Okay, now these T cell receptors, they are formed in the surface of the T cell. Okay, surface of the T cell, but they are not functional. Truly, we cannot say they are truly functional. They again need to be undergo the elimination process, or we can say the selection process. Okay, so that selection process we divided into the events. Okay, so there are 5 events. In the first event, there is the expression of TCR receptor cells. Okay, and they form the 4 types of cells: CD4 and CD8, CD44 and CD24. Okay, now there is then second event. In the second event, there is a selection of beta chain. Okay, so alpha chain is eliminated by the process of apoptosis. So when the beta chain is selected, there is again selection of double positive and double negative charges. Okay, then in the fourth event, there is a selection of positive selection and negative selections, and then in the fifth event, there is a single positive cell selection means they make T cells either as a CD4 positive or they make either as a CD8 positive. See, in the one T cell, all kind of receptor is present. Okay. In the fifth event, what happened? They make them individualized. Okay. Now, as you are knowing that the T lymphocytes, they are classified into the different types, but their basis is their development. Now we see our slides and try to understand the types of T lymphocyte. See the slide. Types of T lymphocyte. So we understood during the processing T lymphocyte are transformed. Understood the word, mark the word. T cells are transformed into the four types. The first one is called the helper T cell or inducer T cells. Okay, these cells are also called as the CD4 cell. Okay, these CD4 cells they are called as the helper T cell. Okay, and in the case of HIV, what happened? There is a remarkable destruction of these T lymphocyte. Or which type of T lymphocyte? These helper T lymphocyte. They destroy the helper T lymphocyte. Okay, the HIV virus, human immunodeficiency virus, they start attacking on the T lymphocyte and capturing their cellular machinery and make them as a viral factory. Not understood? Let me repeat it once again. It's worth to repeat that during the development, the T cells, they transform into the four type. Okay, the first type is called the helper cell, which is basically the CD4 cells. And in the case of AIDS, which is caused by the HIV virus, these HIV virus, they attack mainly on the CD4 cell, or we can say the helper cell. Okay, the helper cells, and capture their nucleus. More specifically, capture their nucleus and make their nucleus to synthesize more viral pathogens. We can say the more viral factory. That's why you cannot directly destroy the viruses. That's why you cannot destroy the virus. If you destroy your viruses, means you are destroying your cell, your major cells. That's a T lymphocyte. You are destroying your major cells. So if there are drugs, they can destroy the T lymphocyte. There are highly potent immunosuppressive drugs, we can say, which can destroy the CD4 cell. But the person who is suffering from the HIV, if their major cells, that's called the helper cell or the CD4 cells, if that destroy along with the viruses, person also die. So any drug which we can say kill the main cells in our body, that are not truly, we cannot say they are a drug, they are the poisons. Okay, that's why HIV are not a 100% curable disease. Okay, they are not a curable disease. Clear? Now, the another type of T lymphocyte which transform during their processing is called cytotoxic T cell or killer T cell. Okay, cytotoxic T cells or killer T cells. These are basically the CD8 molecules. I told you that CD4 or CD8, there are different type of cells, okay, which form during the processing of the T cells into the under the effect of thymosin or thymopoietin in the thymus gland. So they are also called as the cytotoxic cells. Okay, then we have the suppressor T cell and memory cell. Now see the board, I will write down the name of all those four cells. Okay, hope you understood this thing. Now coming to the point, because of the processing, the T lymphocytes basically they transform into
four types. They transform into the four types. The first type is called helper cell. Helper cell, okay, they are basically the CD4 molecules, okay, CD4 cells. Then we have the another cells called the cytotoxic T cell, cytotoxic T cell, or CD8 molecules, okay, CD8 molecules or CD4 molecules. Then we have memory cell, and we have the suppressor cell. Understood? For their formation, all those games is going on in the thymus, okay. So now concluding the today class, we understood that there is a one sequential development of the T lymphocyte, okay. There is a sequential development of the T lymphocytes starting from the intrauterine life, to the fetal life, okay. We understood that there are some of the stem cells, okay, which comes into the liver, okay, the premature livers, and then they migrate into the circulation, then they migrate into the thymus gland, and in the thymus gland, in the cortical area of the thymus gland, under the effect of secretion of some of the we can say the thymus gland cells, basically the epitheliocyte and lymphocytes, two secretions, thymosin and thymopoietin. Under the effect of thymosin and thymopoietins, there is a transformation of these T cells, means they are basically forming the receptors on their surfaces, and they are transformed into the four types. First one is called the helper cell, which is basically the CD molecules. Then we have the cytotoxic cells, we can say the T cells. Then we have the memory cell and the suppressor cells. That's about the T lymphocyte maturation, prolifications, and the transformations, okay. So hope you understood today's class. Thank you with having me. Bye, have a nice day.